All right, Andrew, thank you very much for the introduction, and welcome to everyone joining us today. The title of my presentation is Robust Assay Development for Multiplex qPCR and SNP Genotyping in Miniaturized Volumes on a High-Throughput Instrument. I'd like to start with an outline of the presentation. I've broken it into two parts for today's talk. The first part is going to highlight the use of BHQ Plus probe assays and the IntelliQube real-time PCR instrument for multiplex SNP analysis, and we will highlight examples of data from various research areas. The second part of the talk, I'll switch gears and get into gene expression and the use of BHQ probe for qPCR assay design, and talk about multiplexing considerations and highlight some of the data we've been able to generate. I'd like to start with just a definition of the SNP. A SNP, a single nucleotide polymorphism, is a form of genetic variation where you have a single nucleotide change between individuals or between a pair of chromosomes that would otherwise be identical. And an allele is, by definition, an alternative form of a gene or SNP, and we'll hear this term used frequently throughout the presentation. SNPs are important in a variety of research applications, including human research, where they can serve as biomarkers for disease prediction and are also important in predicting response to certain pharmaceutical drugs. SNPs are also important in the ag biotech sector, where they are used in plant breeding programs for marker-assisted selection or backcrossing, and are also important in trait associations and can be used to track certain animals or plants in these research programs. I will now get into introducing the BHQ Plus chemistry from LGC Biosearch Technologies. BHQ Plus probes have a duplex stabilizing technology that allows them to form stable duplexes with their DNA targets. This leads to enhanced target specificity, which is very critical when looking to discriminate between different SNPs. They are dual labeled, so they have a fluorophore and black hole quencher, and this black hole quencher does not interfere with fluorescent dye signal after hydrolysis. Another nice feature is the real-time design software offered on the Biosearch Technologies website. This allows users to input their target sequences and get assistance in designing BHQ Plus assays. So a typical probe-based SNP assay consists of a forward and reverse primer and two allele-specific probes. These probes are designed to discriminate between the SNP of interest. In this example, we are looking at a C to A SNP. So you would have one probe labeled with a fluorophore specific to the C allele and another specific to the A allele, and this probe would have a different fluorophore. During the PCR process, you get annealing of your primers and allele-specific probes to their intended targets. And through primer extension, the probe is then hydrolyzed by the exonuclease activity of the Taq polymerase. This causes the release of the fluorophore from the quencher and gives you that fluorescent signal that can be detected by your PCR instrument. So using fluorescence-based assays, we can monitor our reactions in real time, but for SNP analysis, it's really that endpoint signal that we are interested in, and that is plotted in a cluster plot. In our example here, we are showing the cow-fluor orange 560 signal plotted in the Y direction, and the FAM signal for our assay plotted in the X direction. So we can use these plots to score our data. And so in the example here, I've highlighted the blue clusters. So these would be samples or individuals that had generated only cow-fluor orange signal, and therefore they are homozygous for the A allele. The middle cluster, or the purple dots, generated FAM and cow-fluor orange signal, and therefore are heterozygous, so they contain both the A and C allele. And finally, the ones that are the red dots here have only the FAM signal and are homozygous for the C allele. So now I'd like to introduce the platform for which this work was done. And this is the IntelliQube real-time PCR instrument. This system is integrated liquid handling, amplification, and detection all in one unit, and is the latest development of the array tape automation. For those of you who aren't familiar with array tape, it is a thin, flexible polypropylene consumable consisting of embossed reaction wells that have a working volume of 1.6 microliters. Now this array tape serves as a replacement for your standard microplates or PCR plates. The tape we are showing here in the bottom right comes as a unique 768 well format. The IntelliQube system has a 5-channel fluorescence detection. The IntelliQube can be hooked up to your laboratory's network and can therefore be accessed by a PC in order to generate your protocols. DNA sample plates are prepared and loaded onto the system and has a 10-plate capacity for sample plates. This can be DNA or RNA samples. It has 3 stations for loading your assay plates, which would contain your master mix and your primers and probes, a touchscreen interface to start your protocols, and after the protocols have run, you can then analyze the results again on your PC. So just breaking that down a little bit more, the first step after starting your protocol, the IntelliQube will dispense your samples. This is an 800 nanoliter dispense. And then your assay, also 800 nanoliters to create that 1.6 microliter reaction. The system automatically seals the array, moves it into the thermocycling station where it will amplify your DNA through the PCR process and collect data in real time. Now this is a continuous process, so as soon as one array is finished, moves out of the machine, the next array is dispensed and ready to go, moves into the thermocycling station, and continues the PCR process. The IntelliQube optics fit well with the proprietary dyes from Biosearch Technologies. These are the CalFluor and Quasar dyes. We have a CCD camera, uses filtered LEDs, and can capture all 5 channels in 15 seconds or less. So when we talk about multiplexing for SNP analysis, we can break that down, and the dyes we use I've broken down in the table at the bottom. For the first SNP, allele 1, we can use FAM, allele 2, CalFluor Orange 560, and our second SNP, in the same well, we can use CalFluor Red 610, Quasar 670, and we use a passive reference, in this case, as TAMRA. And what I'm showing here is the emission spectrum of those dyes, and you can see how those are separated out. So over the next several slides, I'm going to highlight data that we've generated in various research areas and show you the multiplexing capabilities of the chemistry and instrument combination. So for our human SNP genotyping study, we used purified genomic DNA from blood samples, diluted these to 5 nanograms per microliter, and designed assays using that real-time design software. So these are BHQ plus SNP assays. And for this study, we used a clear-call master mix from LGC. We used 4 different SNP assays. These are known to be important for drug metabolism, so variation in these SNPs can impact an individual's ability to metabolize certain drugs. The first SNP is VCORC1, and then there's 3 enzymes from the cytochrome P450 family. So the first slide here showing some data is in singleplex. So all these assays were run, 1 assay per well, and we're showing the cluster plots for the 4 assays. So what we're looking at here again is CalFluor orange signal plotted in the Y direction and FAM signal plotted in the X direction. And we can look at our cluster plots. And what we're looking for here is, do we have good separation of our clusters and good separation as far as signal to noise. So from our NTCs, our no template controls, which are indicated by the black dots in the lower left corner, are we getting sufficient separation? And you can see here we have good clusters, well separated. And what you might notice for this is we only have 2 clusters instead of 3. For a lot of these SNPs, there are rare alleles, so we didn't have any in our population here, any homozygous mutant samples. The next slide is showing the same assays but in a duplex format. So this is running 2 SNP assays in a single well. And so I've broken it down by duplex 1, duplex 2. So for duplex 1, we're running the C9 star 2 and VCore assays. So star 2 was run with the same probes as I showed previously, the CalFluor Orange and FAM. And the VCore C1 in this case was used Q670 and CalFluor Red 610. And what we can see is we were able to maintain very good cluster quality, separation of our clusters in order to make them easily scorable, and we maintained a very good signal-to-noise ratio. Duplex 2, we saw a very similar thing, easily scorable clusters. So just to reiterate, this method is utilizing all 5 channels of our detector, 4 channels for our 4 allele-specific probes, and a reference dye as the 5th channel. The next area where we wanted to demonstrate this capability was for bovine SNP genotyping. For this, we had acquired 29 beef samples from a variety of different grocery stores. Instead of using purified DNA, we used a sodium hydroxide method for a crude prep. And so basically this involves lysing the cells in a sodium hydroxide solution, followed by neutralization in a Tris buffer. We then designed assays, again using the real-time software, and used a clear call master mix for the testing. These SNPs, we dug into the literature and found some various SNPs that were important for commercially relevant traits such as meat tenderness and fat content. The SNPs are listed here with their genomic information. First was leptin, a calpain gene, a calpistatin gene, and a promelanin concentrating hormone. So following the same format from the human data, we ran all of these assays in singleplex. In this case, we did have 3 clusters for all of our assays, but we still maintained very good cluster separation and good signal-to-noise ratio. So all of these in singleplex were just with CalFluor Orange and FAM. Flip to the next slide, and we're looking at the duplex version of these assays, where you can see we maintained very scorable clusters, good separation, and just to reiterate, with the same with the human DNA, we were achieving the exact same genotype calls regardless of the format in which we tested them. The last example, we wanted to show this in plants. So this is with corn or Zea maize. Used heirloom corn seeds, and again a sodium hydroxide prep method to get a very crude extraction of these samples, BHQ plus assays, and clear call master mix. To identify these SNPs, we again looked in the literature and found a publication where they had identified highly polymorphic SNPs, and we picked these not to be associated with any specific traits of interest, purely just because of their polymorphic nature, and we wanted to use them to demonstrate instrument and assay performance. So showing the same kind of format here, these are the 3 assays in singleplex. So this is very crude DNA, BHQ plus assays, and the clear call master mix. We see good cluster separation and a very good signal. I'm going to flip to the next slide, where we show these again in duplex, 843 and 115 duplex together, 176 and 611 duplex together, using all of the same dyes we used before. So our SNP1, CFO and FAM, and SNP2 within a duplex, our Quasar 670 and Cal Red 610. And we were able to maintain our genotype calls between singleplex and duplex, and also very good cluster separation and signal to noise. So with that, I'd like to summarize the first part of the talk. What we're hoping to demonstrate here was successful duplex SNP analysis using the IntelliQube real-time system with BHQ plus assays in array tape. And just to reiterate, these are 1.6 microliter reactions. And we were able to maintain our data quality as documented by good signal-to-noise ratio, cluster separation, and maintaining the calls from singleplex to duplex. And so effectively what this does is allow us to double the number of data points that is achievable with each run. So in our 768 well array, we can really get 1,536 answers from a single array. And we've shown that clear call and BHQ plus probes are an effective combination in a variety of research areas with different sample inputs. So whether that's purified DNA or very crude samples, we are able to see very good data from both methodologies. So I'm going to switch gears a little bit and now get into gene expression using BHQ probes. Just a quick summary of gene expression. It's the process by which genes are transcribed to synthesize functional gene products. Gene expression is important in a variety of applications, including cancer research, drug screening and development, and in agrogenomics. Just the diagram on the right is just showing a very generalized description of how our genomic DNA is transcribed into RNA or a messenger RNA template that is then translated into a functional gene product. There are several methods to analyze gene expression. These include microarrays, RNA fish probes, next generation sequencing through RNA-seq. But what I'm going to focus on for the remainder of this talk is reverse transcription quantitative PCR, or RT-qPCR. So again, just the process of RT-qPCR, we are looking at that messenger RNA, and it first undergoes a reverse transcription step to create the complementary DNA strand, or cDNA, which then undergoes a standard PCR process to get a final PCR product. There are a few different ways we can do this, through either a one-step RT-PCR or two-step RT-PCR, and there are advantages and pros and cons to each of these methods. A one-step process, the cDNA synthesis and PCR occur in the same reaction, so this reduces the pipetting steps, reduces the number of reactions that you need in order to get your answer. However, with a two-step process, the reverse transcription occurs in a separate reaction, so this gives you a little bit more flexibility. If you're looking for many targets from a single RNA sample, you can create that cDNA from that sample. It's more stable, and you can screen many targets against that single sample. So to demonstrate some assay development and multiplexing capabilities of our IntelliQube system with BHQ probes, we wanted to find some relevant targets. So we looked through the data, identified targets that were differentially expressed in various tissues. So we have 3 genes of interest that we'll be looking at today: mucin-1 or MUC1, hepcidin antimicrobial peptide or HAMP, and a solute carrier family 34 member 1 protein or SLC34A1. And we chose some common reference genes: a Tata box binding protein TBP, glycerolaldehyde 3-phosphate dehydrogenase or GAPDH. However, I must note here that there are no universal reference genes, and so the stability of multiple genes should be evaluated for each individual study, and we can't assume because a reference gene is a common reference gene that it's going to work for your particular study. But for our purposes of demonstrating assay design and PCR performance, we chose 2 to work with. And in the absence of having a large pool of samples to choose from, we purchased commercially available human RNA from multiple sources, from multiple source tissues, and designed BHQ assays with the real-time design software. And we decided to use a one-step RT-PCR workflow on the IntelliQube, although both a one-step and two-step process would both work. So I just wanted to walk through some of the assay design steps. First, identified our sequence information. So we're looking at transcript sequences and wanted to design cDNA-specific assays and avoid the amplification of genomic DNA if possible. So we did this through designing exon-spanning assays to hopefully avoid identification or detection of genomic DNA in those samples. The BHQ probes and primers were designed with the real-time design software, and one of the approaches you can take to speed up the design process or the validation process is to just get multiple designs for each assay and test them out initially. This saves time and having to go back and redesign if necessary. Primer considerations. Primers are one of the more important things. We need to understand if our primers that we're going to be used, especially when we're multiplexing, are compatible with each other. So are we forming primer dimers with other assays or within an assay? And there are several software programs available to assist with this. Also, looking at any off-target interactions. So when we're designing these assays, are they specific to our target, or are we going to be amplifying targets that were not intended? And there's several blast programs you can use to use a primer blast to understand what targets am I amplifying. And during the assay optimization process, we always start with understanding what the performance of the assay in singleplex, and then adding assays sequentially when we start multiplexing. So we like to start, after we've designed and acquired our primers and probes, start by characterizing the assay using a synthetic DNA template. So what we're looking at when we're looking for qPCR performance is specificity, sensitivity, and PCR efficiency. And we're looking at gene expression studies. We ideally want to have very similar efficiencies across all of our assays. So we took our synthetic DNA, in this case, and did a dilution series from 10 to the 7 down to less than a copy per reaction, and used 8 replicates per dilution. The more replicates you have, you get a more precise or more accurate answer when it comes to your efficiencies and your limits of detection and so on. For our assays, we use a standard 200 nanomolar probe concentration, 600 nanomolar primer concentration. These can vary depending on the assay or the instrument you're using, but there are standard ranges which are appropriate. So when assessing assay performance, I just wanted to define some of the criteria we're using. Limit of detection, so what is the lowest amount of analyte in a sample that can be detected but perhaps not quantified. Usually this is associated with a stated probability. So assuming a 95% confidence interval, actually the theoretical limit of PCR is 3 molecules. Whereas the limit of quantification, so what concentration can be detected while meeting the defined requirements for error and therefore can be quantified. So a standard method is to use a 35% coefficient of variation. So we're looking at across our dilution series in our standard curve, at what concentration are we exceeding that variation, that 35% CV mark. And then it's the concentration prior to that becomes your limit of quantification. R squared, so how well is our a trend line fitting to the data? So our standard curve, a value of 1, obviously being a perfect fit. And PCR efficiency, which we're calculating from the following formula that's shown. And this is related to the slope of the standard curve, a slope of negative 3.32 equating to 100% efficiency. So this is an example. Before I show all the data, I'm going to start with just gap DH and look at assay performance with our synthetic target. So at this point, it's not RT-PCR. We're just looking at how well our primers and probes working, and we're looking at a singleplex format of this assay. And so in our dilution series, we actually broke it down into 10-fold dilutions down to 1,000 copies, and from there did 2-fold dilutions in order to get a more accurate LOD and LOQ, or limit of detection and limit of quantification. So we can look at our amplification curves on the left, and we plotted our standard curve with quantity along the x-axis and CQ value in the y direction. So now looking at all 5 assays, the 3 genes of interest and the 2 reference genes, we can see very good amplification curves, and the data is actually reflected in the table in the lower right. So we're looking at our LOD, LOQ, R squared, and PCR efficiency. So we can see we had a very good limit of detection, 3 to 5 molecules, and these are estimated copies based on the synthesized synthetic DNA. Remember, with our 95% confidence limit, the theoretical limit of detection was 3 molecules, so we're right down at that theoretical limit. Our limit of quantification was also very good, 15 to 30 copies, R squareds approaching 1, and percent efficiency 96% or above. And typically with PCR assays, generally 90 to above 90% is typically accepted. But again, we want, when we're doing a gene expression assay or study, we want to have all of our assays very similar in PCR efficiency. And just to note here, our LOD and LOQ calculations were performed using a GenX software from MultiD. And the values we're showing for LOD and LOQ are in target copies, not necessarily a concentration, but actual target molecules. So we next all these assays would perform using an one-step RT-PCR process, because it's important to evaluate the performance of your assay in the sample matrix expected for the experiment. So we created a standard curve from 50 nanograms down to 5 femtograms. And on the next slide here, I'm showing the assay performance with the purified DNA in a singleplex format. In this situation, since we have varying levels of gene expression, we weren't looking for LOD or LOQ, but looking at R squared and percent efficiency. And I noted that a few of our assays, since they were lower expressing, we only had 3 points on our standard curve, which is not ideal. Ideally, we like to have more than that, but since due to the low expression, we weren't able to achieve that. So the take home here was that we were able to maintain very high PCR efficiencies switching to the purified RNA sample. So now, getting into the multiplexing considerations. The first thing is we want to make sure we have a master mix that's capable of multiplexing. Not all master mixes are created equal. They need to be formulated with adequate amounts of enzymes, DNTPs, magnesium, etc. It makes sense that we're amplifying more targets. We need to have a sufficient amount of these components in the mix. Primer concentration is very critical as part of the optimization process. Typically, it's a reduction in primer concentration that's going to be necessary, and it's possible too if you have a very highly expressed target. We can limit the primer concentration of that target, and so that's not using up a lot of the materials in your reaction. And like we mentioned before, we start in singleplex and we add assays sequentially in order to see how performance is impacted. And our goal here when multiplexing is to maintain performance as far as sensitivity, efficiency, and similar CQ values. So when I start multiplexing, I want to make sure that I'm getting the same CQ values if I'm running the exact same samples. So for our multiplex example, we chose to use 3 targets and a passive reference dye. So we were utilizing 4 of the 5 channels on our IntelliQube. Target 1 was FAM, target 2, Cal-4 Orange 560, target 3 was Quasar 670, and our passive reference was ROX. So this is an example of how why primer optimization is important. What I'm showing here is an example from our TBP assay. So we're showing amplification curves from a triplex assay, but I'm only showing the TBP curves from that assay. On the left, you'll see when we ran it with a very high primer concentration of the other two assays, the MUC1 and the GAP-DH. We saw at our lower concentrations we had a fanning effect. The curves didn't look good, and we were getting a shift in our CQ values because of it. Now, when we lowered our primer concentrations and empirically determined what was working best, we saw that when we did lower those primer concentrations, we were able to fix those curves. We were seeing better CQ values that were in alignment with what we were hoping to see, and just an overall improvement in signal. The next slide shows the triplex assay performance. So we started with 5 assays, and we wanted to focus in on 3 assays for the purposes of demonstrating multiplexing. So what we're looking at here is singleplex, duplex, and triplex for our 3 different assays, and we can see that the real-time curves maintain their shape. It's going to be hard to really focus in on the performance until I go to the next slide, but just visually we could see we were getting very good PCR performance in a singleplex, duplex, and triplex format. So the table here breaks that down. We're looking at our, again, LOD, LOQ, and percent efficiency in singleplex, duplex, and triplex, with our 3 assays down the left-hand side. So you can see we maintained very good specificity. We did see a slight increase in our limit of detection when triplexing, but there is always something we have to give up when multiplexing, although we were able to maintain our limit of quantification across the board regardless of the plex. And PCR efficiency was maintained singleplex, duplex, and triplex. What we're showing in the bottom half of the slide is just an overlay of the curves from singleplex, duplex, and triplex of our GAP-DH assay. And what we're hoping to show here is that we didn't see a significant change in CQ value regardless of whether we were looking for 1 target or 3 targets in that well. So after we had characterized our triplex assay, we wanted to see if it would maintain performance or we would actually get the same gene expression data using that triplex assay versus if we ran all of those assays in a singleplex format. So we decided to look at MUC1 gene expression in kidney, liver, and lung tissues with the goal of achieving comparable results using a singleplex and multiplex format. We repeated this experiment 3 times in order to assess run-to-run variability of our instrument and the chemistry, and used a comparative CQ approach for relative quantification. This slide shows the data. So we ran 50 nanograms of RNA per reaction, used a 1-step RT-PCR approach, and we're showing the curves in singleplex, triplex, and then overlay of the 2 formats for our 3 assays. And these curves are representing different tissues and sources of these tissues. So we have kidney and lung. And the CQ values we generated from the software were used for our statistical analysis. So first, it's one thing to look at the overlay of the curves, but we want to try to visualize that in another way. So we created a bar chart, and we can see that we're on average maintaining those CQ values regardless of singleplex, duplex, or triplex. And this is shown for the 3 different assays, the MUC1, GAP-DH, and the TBP. So looking at our gene expression results, our 3 goals were to compare between tissues, wanted to compare a singleplex format versus a multiplex format, and also run-to-run comparisons. We used an ANOVA-based approach with a type 3 test of fixed effects, with our effects being the plex, the tissue, and the run. And the only effect that was statistically significant was between tissues, which is what we were expecting. but we did not see a significant difference in our results between singleplex or triplex, or duplex for that matter, and we did not see a difference in run 1, 2, or 3. So to break that down further, we have below the first table, we have the tissue comparison, the mean MUC1 expression expressed in relative log quantities, where we can see the differences in expression between kidney, liver, and lung. And since that was the only effect that was significant with our first test, we broke that down, and the table at the bottom of the slide shows that comparison between kidney and liver, kidney and lung, liver and lung, and all three of those pairwise comparisons were statistically different from each other. And these results matched what we had seen in the publication where we found these assays. So the two graphs on the right are just showing the plexing comparison and the run comparison in mean MUC1 expression. And we can see very little difference, and already showed from the first ANOVA table that we did not have a statistical difference between those. So I'd like to summarize the second portion of the talk. I hope this data has shown you that we are able to successfully multiplex gene targets using BHQ probes and the IntelliQube instrument. And again, just to reiterate, these are 1.6 microliter reactions where we were able to multiplex 3 targets in a single well. And we retain PCR efficiency, sensitivity, and CQ values, which are important when looking at gene expression. And so we saw comparable gene expression results between singleplex and multiplex. And so today we are highlighting gene. There's other applications for one-step RT-PCR, so this could be used for detection of RNA viruses and things of that nature. But the other point I wanted you to take home was that regardless of the instrumentation or the platform you're using, when you're looking to multiplex, assay optimization is going to be a critical step in that process. So with that, I would like to thank those who contributed to this work. At our Alexandria site, Mike Freeman, Alex Kolb, and Andres Rodriguez. At our Hoddesdon site, Laura Hinton and Rebecca Howard. And I wanted to thank Tata Biocenter, Christina Lind, and Vendula Novostova for their assistance in our statistical analysis of the gene expression data.